Okay. I think we're finally ready. Maybe. Okay, so adaptive immune system. So first thing, let's do a quick review of the breakdown of the immune system. How many branches are in the immune system? 2. What are they? Innate and adaptive. So here, I'm going to try and write big so it comes up on the camera. Immune system, 2 branches. Innate and adaptive. Have you guys watched the innate lecture? Anybody remember the mechanisms that we talked about in general in the innate? Barriers, inflammation. Anybody remember any other? Interferon. And there's one more. Complement cascade. Those are the main ways in which your innate immune system protects your body. Today we're talking about adaptive. Let's see, start my slides here. Okay, your adaptive immunity is considered slower. It takes a longer time to act than innate. Because innate doesn't bother figuring out who anybody is, it just simply kills as long as it finds a foreign invader. The adaptive immunity first likes to find out who it is. So it's slower, but it's much more effective. Things that slip past your innate immunity will not slip past your adaptive immunity. Your adaptive immunity has quote-unquote memory, meaning that if you encounter a bacteria or a virus once in your life, your adaptive immune system will always remember that bacteria or that virus. And that memory is what affords you long-term protection. That's where vaccines come in. The vaccines we got as a child still protect us in adulthood. The adaptive immunity itself is a dual system. You have T cells. Why are they called T cells? Because they develop in the thymus, or they mature in the thymus. B cells are called B cells because of the bone marrow. So your immune system has 2 branches, adaptive and innate. Your adaptive system has 2 branches, T cells and B cells. The important thing to remember is even though your immune system has 2 branches, they're not 100% independent of the other. Innate and adaptive work together all the time. Same with T cells and B cells. T cells and B cells work together all the time. What your adaptive immunity focuses on is what's called antigen. Antigen is any molecule that can stimulate a response by the adaptive immune system, by a T cell or a B cell. Okay? It's a very generic definition, and it's meant to be that way. It doesn't say it has to be foreign. It just says it has to be a molecule that can stimulate your adaptive immune system. So this is kind of an overview of where we're going in the adaptive immune system. We're going to talk about lymphocyte development. What's a lymphocyte? Cells that exist in lymph nodes, and specifically name me some lymphocytes. No, not macrophages. What'd you say? Not neutrophils. Those are T cells and B cells. Those are lymphocytes. So keep that in mind. Every time I say lymphocyte, I'm just trying to make a blanket statement to say T cells and B cells. So we're going to look at T cell and B cell, how they develop, and how they do what's called differentiation, which is an important concept. How foreign, or I shouldn't say foreign, how antigens are presented to lymphocytes. And when you present antigen to T and B cells, that's called challenge. So what the challenge of T and B cell lymphocytes does, how they respond. B lymphocytes respond to antigen with antibody, and we'll see how T cells, how they respond to antigen. Because saying that the cells that are with you when you get in injection when you're little, saying that they're still with you 30 years later is theoretical. It's probably true, but maybe it's only one cell left. So they give you a booster to boost that number to make it higher. Okay, an overview of how your adaptive immune system works. Macrophages, which are what kind of cell? Phagocytic cells, okay, meaning that they pick up everything. They pick up parts that are already your body, dead cells, dead proteins, sloughed off cells, plus foreign invaders. And macrophages fall under which part of your immune system? Innate or adaptive? Innate. Macrophage. So this is an example of innate working with adaptive. An innate cell type, the macrophage picks up everything, foreign or self, and they chop it up and they stick pieces of it on their surface, and then they wander over to T and B cells, and they simply ask the T and B cells if they recognize it. T and B cells are going to decide, did that macrophage pick up a dead cell, a dead protein, something self that belongs in the body, or did they pick up a bacteria, a virus, something foreign, something that does not belong in the body? So it's the job of the T cell and B cell to tell the macrophage self or non-self. And self or non-self is determined by MHC. How many people have the same MHC? Only what kind of twin? Identical twins are the only people that would have the same kind of MHC. Every other person, every other anything, cell that exists would have no MHC or a different MHC than you. And that's what T cells are using to determine self from non-self. So how do T and B cells know in the first place what's self and what's non-self? They have been, quote-unquote, trained. And that's what differentiation is. When T and B cells develop and differentiate, they're being trained to tell the difference between self and non-self. So the whole point, and this is a very important thing to remember because you're going to lose it in the muck of where we're going. The whole point of everything we're about to go into is that during B cell and T cell development, we want to produce cells that can specifically recognize only one thing. And we want that one thing to be non-self. Okay? So if I am a B cell or a T cell going through development and differentiation, when I'm done, I will only recognize one very specific substance that's non-self. Maybe I only recognize the cell wall material of a specific bacteria. And maybe my buddy only recognizes the nucleic acid of that bacteria. And then my buddy over here recognizes a totally different bacteria. So if I only recognize bacteria A, and a macrophage brings me bacteria B, I don't recognize it. I don't respond. That macrophage has to continue testing all the different T and B cells until he finds somebody who recognizes what he has. So, starting at the beginning now, you don't want to lose sight of what the whole point is of what we're going to talk about. Both T cells and B cells come from the bone marrow, from that pluripotent stem cell in the bone marrow that we talked about with blood cell development. B cells stay in the bone marrow for their entire development and differentiation before they move out into the general circulation. That's why they're called B for bone marrow. T cells are essentially born in the bone marrow, but for their training, they move to the thymus. That's why they're called T cells, thymic cells. Once they've been trained, both cells will leave the bone marrow and the thymus, and they will migrate to what's called secondary lymphoid tissue. That's the lymph nodes in the body, the spleen, the gulp, the gut-associated lymphoid tissue. They move out into your body in an effort to start protecting you. Okay. T and B cells use over 500 genes to produce these specific receptors that they possess. The way that they use these 500 genes is by recombining them randomly. So, any guesses how many different ways you can recombine 500 genes? It's theoretically infinite, okay? It's a lot. What this means for you is that you can come up with a T cell or a B cell that's specific to every substance known to man, even ones that aren't known to man. There are T cells and B cells in your body that are specific for things that don't even exist because it's a random recombination of these 500 genes. So you are covered. In a fully functioning, healthy immune system, you're covered. Whether you're on this planet or Mars or anywhere else, you are covered for every non-self substance. Because that's what that second bullet point says. Extensive rearrangements over time lead to every possible recombination. Each genetically unique line of lymphocytes, okay, so each specific lymphocyte, when triggered, clones itself. So that very first cell is called the clone. If I have a T cell or a B cell specific to streptococcus and I get strep throat, okay, in order to fight that infection, I have to make a lot. I have to make many copies of that one bacteria or that one B or T cell that's specific for that bacteria in order to fight it. So that's what that's saying. Each line of lymphocytes that came from the original is called a clone. All right. This picture is a visual representation of T and B cell training. So up at the top, we have our lymphoid stem cell right here. This is inside the bone marrow. Doesn't matter T cell or B cell, that's in the bone marrow. That lymphoid stem cell is giving rise to lymphocytes. And we're not even gonna say T cell or B cell, just lymphocytes. Notice that each one is unique, and this artist has given each lymphocyte a different color and a different shaped receptor. This lymphocyte recognizes a yellow circle, this one a blue square, this one a brown triangle, on and on and on. Each one is specific. The first step in the training is that each one of the lymphocytes is presented with self, with material from your own body. For B cells, this happens in the bone marrow. For T cells, this happens in the thymus. If they react to self, if they recognize self as something bad and they begin to activate themselves, they're killed. These two clones, here and here, started to recognize the self molecule, so they were what's called clonally deleted. They were eliminated. And now, we've been developed and differentiated. The remaining lymphocytes are allowed out into the circulation, into the general population, where they will wait until a macrophage picks up an antigen, presents it to them. Red triangle came into your body, a macrophage picked it up. Oh, it presented it to everybody. Nobody recognized it except for this one. This one recognizes red triangle or brown triangle, whatever it is, and it begins to clone itself. In a nutshell, that's how your adaptive immune system works. You make an endless amount of T and B cells that are all only specific for one thing. If that one thing is not self, they are allowed out into your circulation where they await a macrophage to show them what they recognize. Obviously, you're going to go through your whole life with a lot of T and B cells that never get to recognize what they're specific for, and that's fine. As long as it's not specific for you, it's not going to do you any harm for them to be in your circulation. Okay. Clonal selection theory. Lymphocyte specificity is pre-programmed, existing in the genetic makeup before an antigen has ever entered the tissues. That is the theory that I have presented for you. That says that I made specific cells before I ever had the antigen. Somebody who argues against this would say, no. I think that the only reason you get specific lymphocytes is when specific antigen comes into your body first, and then your body makes specific lymphocyte. It is a school of thought, but the vast majority of immunologists agree with what I've presented to you, that it is pre-programmed in the genetic code, and that T and B cells are antigen specific before they've ever seen the antigen. And the research, for the most part, supports that theory. The second part of the theory is that each genetically distinct lymphocyte expresses only a single specificity and can only react to one type of antigen. That's what I've presented you with. Somebody who argued against that would say, no. Once they get out into the circulation, they could switch and be specific for something else. Okay? What I've presented to you says they are, quote-unquote, born with a specificity and they will die with that specificity. And if they never run into that antigen, they will never activate. Again, most of the data supports what I've presented there, but there is a small constituency that's going to argue against it. Okay, a problem that may have already cropped up in your mind, if it hasn't, we have it here. We're relying on random genetic recombination of these 500 genes to give us our antigen specificity for every possible substance that exists. Inevitably, substances that exist include you. So you're also going to generate antigen specificity towards yourself. Those particular clones should be deleted during development. The B cells should be deleted in the bone marrow. The T cells should be deleted in the thymus. Remember that the first step was being marched across self molecules. If you are not aimed towards self, you should ignore it. If through random genetic recombination your receptor was generated to recognize self, and you recognize it, you begin to activate, then you should be deleted. What do you think happens if you're not deleted? What would be the ultimate result of not deleting a self-recognizing clone? Autoimmune disorders. That's one theory of autoimmune disorders, is that for some reason self-recognizing clones slipped through and were allowed into the general circulation. Okay, a little bit more specifically now. The specific receptor on a B cell is called a B cell receptor, or BCR. This is the protein on the surface of the B cell that specifically recognizes whatever antigen it was trained to through genetic recombination. So it's the receptor genes that undergo the recombination to come up with the unique sequence called the B cell receptor. Immunoglobulins. What is the layman's term for immunoglobulin? Do you remember? We talked about it a little bit in blood. What? Antibody. Immunoglobulin is antibody. B cells produce all different types of antibody that we'll talk about at the end of this lecture. But you should know that an immunoglobulin, an antibody, serves as both something that the B cell makes and as the B cell receptor. One type of immunoglobulin is the receptor. A different type is an antibody that the B cell produces. The T cell receptor is very similar to the B cell receptor because it does also have that same 500 set of genes that randomly recombine and produce a specific T cell receptor. But it's a protein lodged on the T cell surface, end of story. It doesn't have a different type of T cell receptor that it makes to fight infection the way that a B cell does. T cell receptor simply sits on the surface of a T cell. Okay. T cell maturation occurs in the thymus. Okay. T cells also have additional proteins on their surface called CDs, clusters of differentiation. And they pick some of these up during differentiation. So along with T cell receptors, you also have what are called CD4 positive and CD8 positive T cells. CD receptors that differentiate two populations of T cells that we'll talk about here in a minute are only capable of recognizing antigen in complex with MHC. MHC is a big part of what T cells do. They will not even approach or even talk to another cell unless it has MHC. There are lots of clusters of differentiation, and more are being discovered every day. But we're only going to focus on two of the very earliest ones, CD4, which are called helper T cells, because essentially their job is to help stimulate other lymphocytes, and CD8, or called cytotoxic T cells. Cytotoxic. Cyto means cell, toxic is going to ultimately lead to destruction or death of a cell. CD8 ones are the ones that do the killing. CD4s are there to help everybody get stimulated and clone themselves. This is a chart I threw in your notes just to help you compare T and B cells, where they develop. They both have CD molecules, but I don't want you to worry about the B cell, just worry about T cells being CD4 and CD8. B cells circulate in low numbers in the blood. When you're not fighting an infection, B cells like to hang out in your lymph nodes and your spleen, whereas T cells like hanging out in the blood. They're receptor for antigen. In terms of requiring antigen presented with MHC, just think of B cells as no and T cells as yes. Everything else we've talked about. So just a little kind of summary chart for you. All right, let's stop and talk a little bit about antigens. Antigen is any substance that provokes an immune response from your adaptive immune system, from specific lymphocytes. Again, it does not say that it has to be foreign. It simply says it has to provoke a response. So how do you get to be an antigen? You have to be perceived as foreign. Doesn't mean you have to be foreign, you have to be perceived that way. How can I be perceived as foreign? Complex composition will do it for you. Having a large molecular weight, being a large protein, a lipoprotein, which is a protein in a fat, a glycoprotein, which is a carbohydrate in a protein, nucleoprotein, which is a nucleic acid in a protein, polysaccharides, which are complex carbohydrates. All of these are very large, very complex molecules, and that's really the key to being perceived as foreign, being large and complex. Here's the molecular weight. Antigens tend to have to be above 1,000 Daltons, which is a molecular measure of weight, in order to be recognized pretty quickly. So what that means for your immune system is that the smaller something is, the longer it takes your immune system to find it. In terms of bacteria and virus, who's larger, a bacteria or a virus? Bacteria. Bacteria are hundreds of orders of magnitude larger than viruses. It's much harder for your body to find a virus, not only because viruses are small, but they hide inside cells. So that's why viral infections tend to knock you down for a lot longer than a bacterial infection. Another important thing to remember is that lymphocytes really only recognize a piece, or what's called an epitope of something foreign. Again, as a lymphocyte, I don't recognize all of Streptococcus pneumoniae. I don't recognize the whole thing. I maybe only recognize the cell wall. My friend over here maybe only recognizes the ribosomes. Another friend only recognizes the nucleic acid, and on and on and on. No lymphocyte in your body would recognize the entire bacterial or viral particle. It has to be a tiny piece of it. So that you end up activating all the ones? Yes and no. They don't communicate directly with each other, but once one gets stimulated, it starts to send out chemicals that make it easier to stimulate anybody else who recognizes it. So here's an example of foreign substances: microbial cells and viruses, foreign human cells, plant molecules. These would be different epitopes. So these 2 orange and 2 green molecules, okay, represent the whole. Your lymphocytes are only going to recognize maybe just this orange one or this green one. Or if this were a nucleic acid, I only recognize blue circle or orange square. Okay? Out of these 3 molecules on the bottom, 1, 2, 3, which one is most likely to be perceived as foreign? 3, the largest and most complicated one. The more parts something has, not only does it make it larger, it also makes it more complicated. So 3 is most likely to be identified as foreign. 1 and 2 are too simple and small. Okay. Antigen processing and presentation. So a macrophage picks up a foreign substance, a bacterial cell. The macrophage can engulf the entire bacterial cell. Thank you. It engulfs the entire bacterial cell, it chops it up into different epitopes, and it puts them on the surface. A macrophage that can do this is also called an APC, antigen-presenting cell. Antigen-presenting cells are mostly macrophages, dendritic cells, which are like a cousin of a macrophage, and B cells. A B cell is a phagocyte. It can engulf, but then it has to present to a T cell. So there's a couple different mechanisms to activate here. We're going to focus on a macrophage presenting to a T cell. So antigen-presenting cells engulf, chop it up, put it on the surface with MHC. Okay. T cell dependent antigens. So that means an antigen that will be recognized by a T cell requires recognition steps. You don't want it to be easy to trigger your immune system because then it might accidentally happen. You actually want it to be hard. You want your immune system to really have to be sure before it activates. So, my antigen-presenting cell picks up an antigen, chops it up into epitopes, puts it on the surface with MHC, and then it will begin to wander around to CD4 cells, T helper cells, and it will ask, do you recognize this? No. Do you recognize this? No. Do you recognize this? No. Finally, somebody's going to recognize it. Okay? That T cell is going to come up. It's going to want to see MHC first because it wants to know, as a macrophage, do you belong here? Do you know the secret handshake? Yes, I know the secret handshake. Don't kill me. But do you recognize what I found over here? So it will check the MHC molecule. It recognizes the antigen. Once it recognizes the antigen, the macrophage says, great, let's get going. It produces interleukin-1, which is a cytokine designed to stimulate the T helper cell. In response to interleukin-1, the T cell will make interleukin-2, which is a chemical designed to go into the system to help the overall activation of other T cells and B cells. That's why this is a helper cell. Okay, so here's a representation of what we just talked about. Foreign microbe engulfed by macrophage, chopped up, processed, and put on the surface in conjunction with MHC. So this orange crab claw is MHC. This little blue peanut is our epitope. So the macrophage is going to wander around to different T cells. Eventually, it will come up to a T cell. Here's my T cell receptor. Here's my CD4. Notice that the CD4 molecule is longer, so it's going to make contact with the MHC molecule first, before the epitope comes in contact with the T cell receptor. So we check it out. Do you belong in the body? Yes, I do. Great. What do you have? I have this blue peanut. Do you recognize blue peanut? I absolutely do. I've been specific for blue peanut my whole life. Perfect. I'll give you interleukin-1. You release interleukin-2, and that's going to start a notification to your immune system that you've been invaded by something foreign. And at the same time, there are thousands of other macrophages that have found it. They are finding other T and B cells that recognize other parts. You're all kind of beginning to get stimulated at once, and it's kicking off your adaptive immune response. Okay, B-cell activation, which is going on concurrently. So B-cell activation also requires a sequence of events, and we don't want it to be easy. Clonal selection, which means the macrophage or the T helper cell, somebody came up and said, you, B-cell, we need you, you're specific for this antigen, binds the antigen, other chemical mediators, other interleukins occur, then a signal is sent straight to the B-cell nucleus, and it says, begin to transcribe and translate antibody. Okay? Also, I need more of you. So you need to undergo mitosis, which means copies of yourself. The B cell is going to clone itself into two different populations, what are called memory cells and plasma cells. Okay, memory cells and plasma cells. Memory cells are simply designed to last a long time. They're not fighters. Plasma cells are your fighters. Those are the ones that are designed to make antibody. And to give you an idea, a plasma cell, once it starts making antibody, can secrete 2,000 molecules of antibody every second, okay, that it's activated, which is a lot. Antibody is the same as what other molecule on the B cell? Immunoglobulin, but something more specific. The B cell receptor. I want you to just take a minute because this should make very intuitive sense. If what makes you specific is your B cell receptor because it recognizes an antigen very specifically, it's a pretty nifty mechanism to be able to make copies of your specific B cell receptor, but then let them go out into the body. Because they are only then going to recognize what? They're going to only recognize whatever it was that B cell was specific for. So if I'm a B cell, and my B cell receptor is specific for the cell wall of Streptococcus pneumoniae, and I have a mechanism whereby I can make copies of my B cell receptor and let it go, then my antibodies are going to go out and they're going to start to cover the surface or the cell wall of Streptococcus pneumoniae. And we're going to see how that helps your immune system. But the more specific the antibody is, the more tightly it binds. So by having this mechanism where you were specific in the first place and then you let go of your antibody, you can be assured that when they find their target, they're going to bind very, very tightly to it. Here's B cell activation. Okay, in this instance, the B cell engulfed by itself. It served as the antigen presenting cell. It went to a T helper cell. There was recognition. Then the B cell activated memory cells, plasma cells. The plasma cells begin to secrete antibody, and the antibody goes out and finds the original microbe that was engulfed by the B cell. Okay, again, what does antibody do? Antibodies are designed to bind specific antigens because they're a copy of the B cell receptor, and that's what the B cell receptor does. Antigen-antibody complexes are characterized as weak or strong, how tightly they bind to each other. If it's a copy of the B cell receptor, it should be very strong. 4 things occur when antibody binds to antigen: opsonization, neutralization, complement fixation, and agglutination. So I think this picture is the best. In the upper right-hand corner here, you have opsonization. Remember we said that small things are harder to find? So if it's a small thing, it's hard to find. But if it's a small thing covered in antibody, that's like shining a flashlight on it for your immune system. It makes it very easy now for macrophages or other even granular leukocytes to find it, engulf it, and kill it. That's what opsonization is. Makes it easier to find and engulf. Neutralization works well with something like a virus. If you watch the interferon part, of the innate immune system, one of the things I kept saying was that you cannot block a virus from entering a cell. Antibody can. Antibody can cover the surface of a virus so that it is blocked from entering a cell. So that's one way it can neutralize. Agglutination is another help for things that are small, making them larger. Agglutination is when you take several small particles and you stick them together. The glue that's holding them together is antibody. Okay, so if I have one small particle, it's hard to find, but if I stick it to 10 others, now I've got a giant chunk. And the glue that's holding it together is antibody, and that's what agglutination is. The last one, complement fixation. Anybody want to take a stab at where antibody fits into the complement cascade? We used a very vague term when talking about the complement cascade. It was at the very beginning. The initiating molecule. I just simply said in that lecture, hey, there's an initiating molecule and then the complement cascade gets going. The initiating molecule is antibody. So this is another crossover between the innate and the adaptive. The plasma cells help start the complement cascade in the innate immune system. Five classes of immunoglobulin or antibody that are called isotypes. IgA, IgM, IgG, IgD, and IgE. And probably at one point in my educational career I knew what those letters were for. I don't know anymore. IgG, when we talk about antibody and antibody used to fight infection and what kind of antibody is circulating in your blood right now, and if I drew your blood and looked for antibody, we're always talking about IgG in those instances. This is the primary antibody type used to fight infection, and it's the most numerous. If I drew your blood looking for all the different types, I would find by far the most IgG. IgA is in your mucous membranes, it's in your tears, it's in your saliva, it's in colostrum. What's colostrum? It's the first milk that comes from the breast of a mammal. Okay? Within the first 24 hours, the mother secretes colostrum. Very thick, high fat content, high antibody content, because theoretically, and there's lots of arguments for this one, when the mother and the infant are close to one another physically, they should be getting exposure to the same bacteria, viruses, foreign invaders. Mother makes the immunity, passes it to the baby through the breast milk. That's what colostrum is for. That's where you're getting IgA. It's passing immune substances to a newborn. IgM. When a B cell is first triggered into a plasma cell, before it makes IgG, it's going to spend a few minutes making IgM. IgM is the first isotype synthesized so that it can get the message out to your innate immune system and get that complement cascade going. Then it does what's called isotype switching, stops making IgM, starts making IgG. IgD is the one type of antibody that's stuck. It can't go anywhere because it is the B cell receptor. And every B cell doesn't just have one receptor randomly on its surface. It's covered with its own B cell receptor. But IgD is stuck on the surface of the B cell. IgE is your worst enemy if you have allergies. IgE stimulates inflammatory responses, especially the ones that surround allergy, anaphylaxis, asthma. All of those are made worse by IgE. This is just a chart showing you the different isotypes. They are structurally different as well as have different functions. Okay, primary and secondary response to an antigen. Primary is what type of response in a timeline? First, and then secondary would be second. So what this is saying is, what's the difference between the first time I see an antigen and the second time or the third time or the fourth time, any subsequent time I see the antigen? Due to the presence of memory cells, the immune system responds differently the first time than it does any other time. The first time is characterized by a latent period. Latent means waiting and nothing's happening. Okay? Latent period is when you have been invaded by the bacteria and your macrophages have picked it up and they are desperately trying to find the T and B cells that recognize it. But remember, if you've never seen it before, you maybe only have one or a small handful that recognize that. In the billions of cells in your body, maybe you have a dozen that recognize your new foreign invader. So it takes a long time. Days, usually 3 to 5 days. This is when you feel horrible, you're running a fever, you're getting your sore throat, you're having your symptoms, you're vomiting, any of those kind of sicknesses that are coming up, because nobody can really kill it. It's multiplying inside of your body, trying to take over your resources. Your innate has already failed. It obviously got in through a barrier. You are inflamed if you have a fever and other vascular changes. If it's not a virus, interferon's not involved. And remember, the complement cascade can't start until the innate or the adaptive immune system makes IgM. So if it got by the barriers, you are just having to wait until your adaptive immune system kicks in. And that's what causes you to feel bad. Once it finds it, then B cell activation, T cell activation, clonal expansion occurs. That's what makes your lymph nodes swell. Because when T cells and B cells clone themselves, essentially the Xerox machine is the lymph nodes. They migrate into the lymph nodes, make copies of themselves, and then extend out. It's not the bacteria that has invaded your lymph node, it's your T and B cells clonally expanding. You make all these cells, you make all the antibody, all the cytotoxic T cells, you kill the bacteria, you feel better, your fever breaks, great, wonderful. The second time you ever see that bacteria, chances are you won't even know that you ever encountered it again. You won't feel bad, you won't get tired, you won't throw up, you won't run a fever, because the second time that bacterial cell comes into your body, or that viral particle, it comes in, it's engulfed by an antibody, or it's engulfed by a macrophage. Now, instead of looking for a dozen cells out of a billion, now I have probably tens and thousands of cells from the memory populations from the first time I saw that bacteria. So we find it quickly, we kill it before it has a chance to multiply inside the body. All is well and good, and you don't even get sick. Yes? Usually the only time you can pass it is during the latent period. And sometimes during the latent period, you don't even begin to feel bad yet. You're harboring, it's multiplying inside of you. That's the time when you're most likely what's called shedding virus. But once you start to feel bad or are coming out of it, you don't generally pass it on anymore because your immune system's got it under control.
But once you kill it, you get it back?
No. Well, you can. But chances are you won't ever recognize it. The reason that every year you get a cold and every year you get a flu is because it's a different virus. Cold viruses and flu viruses are what are called retroviruses, and they mutate like crazy. A lot of times the one that comes into you doesn't even look the same when it leaves. It mutates inside your body and leaves. So that's the reason why you can get it so many times and you feel like I just had a cold or I just had a flu and I'm getting it again. It's all different. You're running into a different virus technically every time you get it.
When they tell you, like, children are on fever, keep them home, they run a fever, is that because the initial
Yeah, you're still in that latent period. They can still be shedding virus. Yeah, the safest time when you're not probably shedding any more virus or any more bacteria is, of course, when you start to actually feel better. That means your immune system has gotten a hold of whatever it was and neutralized the problem, is just cleaning it up now. This is a picture of what happens to T cells. It's essentially very similar to what happens to B cells. The two panels represent CD4 cells and CD8 cells. The top panel is a CD8 cell, which is a cytotoxic T cell. It recognizes the antigen brought to it by the macrophage. It activates, and then it clones itself into two populations, memory cells and activated cells. Activated cytotoxic cells go after the foreign molecule with what's called a preferin. Anybody want to guess what a preferin does? What's a perforated page? What does it have in it? It's got holes in it, so it makes it easier to rip. Preferins are molecules that eat holes in cells, which again, if you haven't watched the innate immune system, that's one of your immune system's favorite way to kill things, just to poke holes in it. Preferin is another molecule that accomplishes that. CD4 cells, the T helper cells, same thing. They activate, they clone into two populations, memory cells and activated cells. CD4 helper cells run around and they help activate macrophages and B cells, and they help get that immune response going. Okay, stepping back now and looking at your adaptive immune system as a whole, talking about the different types of immunity you can be afforded. Active immunity and passive immunity. Active immunity is what happens when you receive an immune stimulus and you go through everything that we just talked about. Macrophages present, cells activate, they clone themselves into two populations, memory and activated cells, you fight the infection, wonderful. You are afforded long-term protection because you made memory cells, you probably experienced a latent period where you didn't feel very good, and active immunity is only achievable by people that are immunocompetent, meaning you have a healthy, fully functioning immune system. Passive immunity is what happens when somebody simply receives an immune substance. This can be most often antibody. If I just give you the antibody to whatever it is you're fighting, but people also receive interleukin as a treatment, IL-1s and IL-2s in an effort to help them. In terms of passive immunity, you didn't have to make the antibody. So you don't have any memory. You didn't make any of your own antibodies. You're going to get immediate protection, but as soon as your liver metabolizes those antibodies, you're done. It's a short-term solution. Natural and artificial immunity. Natural immunity is what's achieved without medical intervention. If I run into measles, mumps, and rubella on my own, and I live, slim chance if I ran into all three of them, if I lived, I would have natural immunity to those foreign substances for the rest of my life. But I have artificial immunity to those particular foreign substances. Medical procedures were used to aid in my immune reaction to them. I was injected with killed or weakened measles, mumps, rubella, and I made my own response to it without dying, without having a latent period. That's what a vaccine is. Yes?
So the natural immunity, is that like when they say you have chicken pox, but yet you get chicken pox again?
Natural immunity to chicken pox would be you got it when you were little, you had the itchy rash and everything. Artificial immunity to chicken pox would be getting the vaccine for chicken pox.
So when the natural immunity is like you get it when you're little, and you have it, then you get older, you have it again.
Yeah, and it manifests as actually a separate disease, shingles, because, and don't quote me on this, I only think this is right, varicella zoster, which is the virus that causes chickenpox, harbors itself in your spinal column. And it should never come up again, but for some people, and I don't even think they know what triggers it, and some people it just comes awake again. So your body doesn't actually fight it, it kind of hides it. And it should never come out again, but in some people it does.
Because I was diagnosed with chickenpox.
Shingles, yeah. For whatever reason, the virus keeps reappearing in your body. Yeah. Yeah, chickenpox is a weird one. I don't know completely all the information about that virus, but you're right. It can come again, even if you've had natural immunity to it. Okay, passive immunization. This is mostly receiving antibodies. Immune serum globulin. That means I took your serum, which is the liquid portion of your blood, and I took all the immune substances out of it, which is essentially the globulin, the antibodies. So you get a concentrated dose of antibodies administered to you, and the antibodies enter into your system and they eliminate potential pathogens. This is a popular treatment for hepatitis B and as well as for HIV. One of the more interesting ones is any kind of venomous anti-serum. Like snakebite anti-serum is made in horses. The reason it's made in horses is because we need it mass-produced. Horses have a lot more blood than you or I, so they have a lot more serum that they can lose without damage to their body than you or I. So I put a small amount of snake serum into a horse. It makes the antibodies. I harvest the serum. I ship it to the doctors. Here's the interesting thing. I get bitten by that snake. I go into the doctor's office, and I say I've been bitten by the snake, and he gives me the horse's antibody. It goes into my body. It eliminates the threat, the venom that was floating around in my blood, and my body says, thank you very much, but that was the weirdest antibody I ever saw. It was definitely something strange about it. Let's go ahead while nobody's looking and make some antibodies towards that antibody in case we ever see it again. You go into the emergency room because you got bitten by that snake a second time. If they re-administer the horse antibody to you, it's most likely going to put you into some kind of weird anaphylaxis because your body recognized that antibody as made in a different species. There was something not right about that antibody. So how do you get around it? If you get bitten by that same snake twice, next time you have goat anti-serum, and the third time you have sheep and dog and cat and human and every other type of anti-serum you can have without having the same one twice. Protection when you're given an antibody is going to last 2 to 3 months depending on the dose you were given and how fast your metabolism is. Active immunization is what a vaccine is. The key to any good vaccine is to be antigenic but not pathogenic. Explain to me what that means. What is an antigen? Definition. Substance that generates a response by your adaptive immune system, your lymphocytes. What's a pathogen? Causes a disease. So what that statement says is, I'd like to provoke your immune system, but I don't actually want you to get sick. I want to give you immunity to chickenpox, but I don't want you to get chickenpox. That's the key to any good vaccine. Antigenic, but not pathogenic. So the antigen that's selected for a vaccine has to be effective, which means antigenic, not pathogenic. It has to be easily administered. It has to be safe, meaning doesn't cause the disease, and also cost effective. So you have to meet kind of all of those things before you can release a vaccine. We want the same response you would get as if you saw that foreign substance all on your own. We want your body to freak out, we want it to activate, we want it to make memory cells and plasma cells and do everything it would normally do, just a little bit less intense. The big thing you want are the memory cells. That's the number one thing you want from a vaccine. Different vaccine preparations killed whole cells. So instead of you seeing the live bacteria by yourself, I killed the bacteria and then I injected it into you. A lot of times your body will still pick it up, chop it up, present it, see it as foreign, you get a response. Inactivated viruses. I've done something to the virus to make sure it cannot replicate inside your body. Live or attenuated. What does attenuated mean? Weakened. It means weakened. Attenuated means weakened. Sometimes I do give you a little live cell. Sometimes it's alive, but I've weakened it. Okay? Sometimes I don't give you the whole thing. I already chop it up, and I just give you antigenic molecules from the bacteria or the virus. And then more and more we're seeing some type of genetic engineering of viruses, of microbes, or microbe parts or agents. Killed or inactivated cells or viruses have usually been subjected to heat, radiation, or some other agent that killed it or weakened it, but not so far that your body won't recognize it as foreign. If you completely obliterate a foreign substance, sometimes your immune system won't see it. It won't pick it up or perceive it as foreign. So you have to preserve that antigenicity. Live weakened microbes usually come from modified growth conditions, usually starvation. Whatever it is that microbe needs to survive, I didn't give it to it. Okay? I kept it alive, but barely, through starving it. And then I pulled it and injected it into you so that it was too weak once it got into your body to multiply and really take over. Live vaccines are by far the best in terms of immune response. They require few doses. So if you ever look at a child's vaccine schedule, you'll see some vaccines require 4 and 5 boosters. Those are the dead ones, okay? Those ones, you need more and more exposure to them. Some only require 2. That's probably a live or genetically engineered one. So live ones give you the greatest immune response, fewest doses, but are not always cost effective because whatever company made that vaccine had to pay to keep that virus alive or that microbe alive and multiplying, which requires special storage. You have to worry about the fact that it could be transmitted to other people, people that work for making that vaccine. Also, people that receive that vaccine have the possibility of transmitting it. It could mutate back to a virulent strain, which is a disease-causing strain. So let's say I genetically engineered a virus and I took out whatever made it cause disease, but I kept it alive. Well, what's saying I don't inject it into somebody, it just mutates back, and all of a sudden it causes disease again. So you have to take into consideration, if you're a pharmacy company, all of those things. Examples, killed or inactivated is here at the top. Get my mouse going. So it's alive, I use heat or chemicals. It's dead, but it's still intact, so it's going to stimulate an immune response. Live or sometimes even attenuated go in, and the best part is that they can multiply a little bit. Multiplying inside your system really freaks your immune system out. It finds it faster, it makes a more robust response. Those are whole cell vaccines. Subunits. Okay, here is a virus particle. Maybe I just take a part of the cell wall. Maybe I just take the proteins that appear on the surface of that virus. Or if this is a bacteria, again, maybe I take proteins on the cell surface, the peptidoglycan, maybe I take the nucleic acid, maybe I take the flagella. It's like putting the microbe in a blender and then giving you the parts. And then lastly is genetic engineering. So there's a virus that has a specific surface antigen, but it's so virulent, I don't even want to mess with chopping it up and giving it to you. So I go into that virus's DNA, I find the part that makes that surface antigen, I cut it out, essentially you give it to a yeast cloning vector, and it just kind of stupidly makes whatever DNA you give it, so it continually makes pieces of hepatitis B's surface antigen, spits it out, you harvest it, and put it into a vaccine. The last thing I want to talk about in the immune system is immunopathology. Pathology, again, is disease. So these are diseases of the immune system. Diseases of the immune system are associated with one or two things: underreactivity or overreactivity. Your immune system either did not respond strong enough or at all, or it over-responded. Allergies, anaphylaxis, grafts, and transfusion are all overreactions. Immunodeficiency is an underreaction. Here's a breakdown of the different things. On the left-hand side of the screen, we have underreaction, lack of surveillance by the T cells and B cells, or other various problems can lead to cancers and also immunodeficiency. On the right side of the screen, we have overreaction. Allergies and anaphylaxis break into 4 groups: type 1, type 2, type 3, type 4. Types 1, 2, and 3 are mediated by B cells. It's an overreaction of a B cell. Type 4 is an overreaction of a T cell. So that's what we'll talk about first. An allergy is a condition of altered reactivity or exaggerated response by the immune system. It manifests itself with inflammation. So it's not only the adaptive immune system that's playing a role. The adaptive will also pull in the innate with inflammation. The antigen that causes an allergy is called an allergen. It's being perceived as foreign, even though it's not there to do you any harm. Again, 4 levels of hypersensitivity or allergic state: type 1, type 2, type 3, type 4. Here's an overall chart. If you want a summary, we're going to talk about all of this, but if you wanted to just go back and get a quick review, that's what this chart's for. So type 1 is called atopy and anaphylaxis. Type 1 is characterized by having an immediate onset, okay, which means you experience your symptoms right away. You see, it requires exposure to the specific allergen, so it's not just randomly set off. You do see your allergen. The difference between atopy and anaphylaxis. Atopy is a chronic local allergy. Chronic means long-term. Local means confined to one area. So this is like hay fever or asthma. Asthma is confined to your respiratory system. Hay fever is usually confined also to your respiratory system. Your nose, your eyes, your face are most often what's affected. Anaphylaxis is systemic. That means the entire body, most importantly and dangerously, your cardiovascular system is responding to the allergen. Anaphylaxis can be fatal due to airway obstruction. Okay? If your respiratory system and your cardiovascular system are both involved, that can lead to fatality. You can obstruct your airways and circulatory collapse. The epidemiology of allergies. Epidemiology is how anything affects a population. 10 to 30% of the population suffers from atopic allergy. Is that number underreported? Highly likely. Because for most people that have just a minor allergy, drive down to the drugstore, get some Claritin, get some Zyrtec. Never tell your doctor about it. Okay? So the only way we have these numbers is because somebody went to a doctor's office and sorted through the files. If you never told your doctor you had an allergy, you're not a part of that number. 35 million people who suffer from hay fever spend about $500 million on treatments, over-the-counter treatments. So that's why every 10 seconds you see an allergy commercial on the TV, because this is a big money business for pharmaceutical companies. Familial, which is a hereditary association, is only a susceptibility, not the specific allergy. So let's say I'm allergic to peanuts. Okay? Does that mean my daughter is going to be allergic to peanuts? No, not at all. All that means is that I'm allergic to peanuts because I have too much IgE. That's usually what causes an allergy, is too much IgE circulating in your system. The only thing I've passed on to her is the probability that she's also going to produce too much IgE. And it's going to manifest itself on shellfish or strawberries or some other highly allergic substance. If I'm allergic to peanuts and my daughter is also allergic to peanuts, that's a coincidence. She just happened to manifest her allergy onto peanuts as well. I did not pass her that specific allergy. Modes of entry for allergens. Respiratory tract is a big one. Gastrointestinal tract for food allergies. Skin. Skin can be tiny breaks in your skin. It can find its way in through glands, even through hair follicles if it's small enough. A very important thing to remember about allergies is the organ of allergic expression may or may not be the same as the organ that let the allergen into the body, portal of entry. What that means is that I may have eaten something. Okay, let's say I have a shellfish allergy and I had some shrimp. So the portal of entry is my gastrointestinal tract. But then I get asthmatic symptoms. My bronchial tubes constrict. I get airway constriction and inflammation. The organ that responded to that was my respiratory system. Now, I could have eaten shellfish and gotten horrible gastric cramping, vomiting, and diarrhea. In that case, the portal of entry is the same as the organ of expression, but it doesn't have to be. So that's important to keep in mind, especially if you're an allergy sufferer. You could have eaten something and then a couple hours later gotten a rash. If you get a rash, it doesn't mean something touched your skin. You could have eaten it, you could have inhaled it, okay? It could have come into your body a lot of ways. Types of allergens are basically broken down into how they come into the body. Inhalants are things that come in through the respiratory tract. Pollen, dust, dander, fungal spores. Ingestants are things that come in through the GI tract. Contactants are things that touch the skin. And injectants are things that get injected underneath the skin. Common injectants are side effects of drugs. When you get that quick, low voice, may cause this, and this, those are side effects of a drug, and those qualify as an allergic reaction. Again, more examples. Under injectants, that also includes venom and bee stings. Those are also types of injectant allergic reactions. Okay, type 1. In general, not always, type 1 allergies develop in stages, which means the initial encounter with whatever the allergen is what's called a sensitizing dose. It comes into your body is very disturbed by it, but it just makes the memory cells and it sits and waits until the next time you see it. Subsequent exposure can lead to the allergic reaction where you get itchy eyes, runny eyes, and swelling up, hives, whatever you get. This is the reason why some people say, I've always taken this, or I've always used this, but all of a sudden I'm allergic to it. It doesn't have to be one time, it can be 20 times. Nobody knows exactly how the sensitizing dose works. Why can I take an antibiotic ever since I was a child, and then when I'm 30 years old, all of a sudden I'm allergic to it? Well, the whole time before I was 30 years old was my sensitizing dose, and finally my immune system had enough, and it was going to start reacting to it. So that's how type 1 allergies develop. IgE is involved with type 1 allergies. So once an allergen enters into the system, the B cells will recognize it. They will begin to produce IgE. IgE has a high affinity for basophils and mast cells. What's inside of a basophil? What are the granules inside of a basophil? Two H's. Histamine and heparin. Okay, so histamine and heparin, mast cells have some even nastier substances. So here's an example. Allergens come in, recognized by a B cell. The B cell begins to make IgE. IgE sticks to the surface of basophils and mast cells. Time elapses. The second time the allergen comes into my body, it binds to the antibody, which is on top of the basophil or mast cell, causing it to let go of whatever is inside of it. That goes into my blood cell or my bloodstream, and I begin to get allergic symptoms. Itchy red eyes, what's called wheal and flare, which is hives, runny nose. All of that is being created because your allergen has triggered your mast cells and your basophils to release their granular substances. No, this is a good question. Why are you even having a response? What's the difference between a piece of plant pollen and a bacterial cell in terms of when they come into your body? Why should your immune system react to a bacterial cell, and why should it not react to plant pollen? They're both foreign. No. What's the bacterial cell trying to do to your body that plant pollen is not? It's trying to take you over. A bacterial cell is trying to multiply inside your body and take over all your resources for itself, or a virus, or anything else that's quote-unquote alive. Plant pollen, if that's a mistake, it didn't mean to be there. You can't pollinate a flower inside your lungs or your skin or anything. It's not going to do you any harm. So technically, we don't want your immune system to respond to it, but it is foreign. Your immune system did respond to it, and it perceived a threat, so it makes the IgE. It does clear it out, but the allergy will never go away because of the specificity, because now we have memory cells. Now we're ready to see it again, and we're ready to react to it again if we ever do. It's a type of mistake by your immune system, but when you boil it down to the way your immune system works, it is a foreign molecule. Okay, so it's not that surprising that it recognized it, but you don't need that type of robust response because the plant pollen is not there to colonize you and take you over.
The response to a bacteria is an effort to rid your body of the bacteria. The response
Yeah.
to the allergen, it's not getting, when it responds, is it getting rid of that pollen? Or
Yes, yeah, it's still, the whole inflammatory response is there to help bring more resources in to take care of what it perceives to be a threat. Yeah, so all the same things are still going on that we talked about. You're still getting cytotoxic T cells. You're still getting antibody opsonization, agglutination, neutralization. You're still getting all of that. But in addition, we're getting this reaction between IgE, basophils, mast cells. Other particles, other symptoms of allergic reaction, prostaglandin. Prostaglandin can come out of a mast cell. It dilates blood vessels, constricts bronchial tubes. Also, if you manifest a headache, that's prostaglandin. Histamine, serotonin, bradykinin, those are ones that will cause hives, dilated blood vessels, constricted airways. Also cause your glands to start weeping clear fluid. Leukotriene is a big target by asthma and allergy companies. Because it causes constriction of bronchioles, also mucus buildup. And that's what causes airway obstruction. If I tighten down all my airways and fill them up with mucus, it doesn't take a whole lot to figure out that you could plug it up and not be able to breathe. Circulatory collapse. If I had anaphylaxis and I had a chemical in my body that's dilating all my blood vessels at the same time, get me this next step to circulatory collapse. Why is that bad if I dilate all my blood vessels at the same time? In general, take stroke volume, cardiac output, blood pressure. Your blood pressure plummets. That's what circulatory collapse is. If you had a systemic chemical dilating all your blood pressures or blood vessels, it would drop your blood pressure suddenly, and that's what circulatory collapse is. Type 1 diseases, allergic rhinitis, which is the official term for hay fever, seasonal reaction to plant pollen. Eczema is a type 1 allergic reaction. Eczema is an intensely itchy inflammatory condition of the skin. Again, eczema does not have to manifest because you touch something with your skin. You could eat something and get eczema. You could inhale something and get eczema. Food allergies, drug allergies, these all count as type 1 diseases. Anaphylaxis, again, atopy is what we were just talking about. Anaphylaxis, cutaneous and systemic. Cutaneous is anything having to do with the skin, okay? So cutaneous anaphylaxis would be wheal and flare, which is hives, an inflammatory reaction to a local injection. Generally, it's widespread. Cutaneous anaphylaxis is not local. It's pretty widespread across the skin. Systemic anaphylaxis is the really dangerous one. That's the one that can cause respiratory and circulatory failure that can lead to a fatality. It usually requires a higher concentration of chemicals than ATP. So let's say I had a peanut allergy and I accidentally ate food that had been prepared in conjunction with peanuts, and some of the peanut oil and peanuts were in my food. I would probably have an allergic reaction. If I ate a big old handful of peanuts straight into my digestive tract, that could possibly cause anaphylaxis. A higher dose of whatever your allergen is usually required to trigger anaphylaxis. If you are going into untreated anaphylaxis, you can die within 15 minutes due to airway obstruction with the tightening of the bronchioles and the buildup of mucus, as well as that circulatory collapse from dilating your blood vessels. For systemic anaphylaxis, it depends on your level of reactivity. It's rare that somebody could just be around it and go into anaphylaxis, but I've actually seen it happen before. A woman was having anaphylaxis to something totally different, and the medics came in and touched her skin with latex, and she got even worse right then. Yeah. It depends on the severity of your allergy. Whether even, sometimes just touching it could put you into anaphylaxis. Sometimes you'd have to eat it, roll in it, rub it on you, and then you could go into anaphylaxis. But it just depends on your level of allergy. How do we diagnose allergies? Most popular way is a skin test. When you get a skin test, it's usually on your forearm or your shoulder blade, and you are injected, scratched, or pricked with pure allergen extract, and then you have to wait a certain period of time, and they're looking for wheal and flare, which is redness and raised inflammation. This person right there, a different allergen extract is at each side of those lines. You can see the lines farthest from us didn't give us any reaction. The lines closest to us did. But it's a great way in a very small area to test probably for 20, 30 allergens all at one time, figure out what you might be allergic to. Therapy for allergies, antihistamine, okay, goes in and blocks the histamine that's being released from the basophils. It can't stop the basophils from releasing the histamine, but once it's out, it can tie it up so it doesn't have an effect. Desensitization or hyposensitization, nobody knows exactly what the mechanism is here or why it works, but sometimes repeated exposure to low doses of the allergen can stop you from reacting to it. A lot of people that handle snakes and spiders and venomous creatures go through desensitization, where they're constantly giving themselves a low dose of venom. That way, if they're ever bit, they don't go into anaphylaxis. And for some substances, it seems to work. Others, it doesn't. Why it works, we don't really know. A very common one for severe types of allergies is that passive immunity. Being administered antibodies directed at IgE. You have too much IgE if you have severe allergies. So I administer anti-IgE antibodies. It's an antibody designed to go after an antibody. When the two of them stick together, it neutralizes the IgE that's circulating in high numbers in your blood. Remember that passive immune substances are a temporary short-term solution, only good for 2 to 3 months. So if you were undergoing this treatment, you'd have to go back to the doctor so many times a year to keep having them administered to you.
So which one would that be for, like, going for allergy shots? If you're like allergic to an animal, you have allergy shots, which one of those would it be?
It depends on what you go through. There are some allergy shots that are specifically tailored to go after an antibody for your allergen. So like if you are allergic to dog dander or something, they can send your blood away and a lab can produce a panel of antibodies that neutralizes your specific dog antibodies. They give you a shot, it ties up your antibodies for a while so that you don't react to it.
So it would be actually one of those that you can say, the
Desensitization would be taking the dog allergen yourself. Would be actually taking the straight dog allergen, the dander or the hair or whatever saliva that it is. The blocking antibodies is a passive immune substance that's being administered to block your reaction. Type 2 hypersensitivities are also B-cell mediated. Type 2 reactivity are reactions from B cells that lyse cells. They literally explode cells. And that is through the complement cascade. This is essentially what happens when you would be transfused the wrong blood. If I'm type A and you transfuse me with type B blood, my body is going to use the complement cascade to destroy it. It's going to lyse all those red blood cells. Okay, so if you have a bad transfusion, you are going to be using the complement cascade. This is also the reaction of hemolytic disease of the newborn, which we haven't talked about yet because we never got to talk about blood types. Hemolytic disease in the newborn has to do with Rh factor, which is what makes your blood positive or negative. So you could be AB positive or you could be AB negative. It's the presence or absence of what's called rhesus protein. If you're positive, you have it. If you're negative, you don't. If a woman is Rh negative and she is having a baby with an Rh positive father, the chances are pretty high that she could be an Rh negative woman carrying an Rh positive baby, which means her antibodies could go to the baby, recognize the baby's blood as foreign, and destroy the baby's blood. Okay? That's hemolytic disease of the newborn. Generally not a problem on the first pregnancy because the placenta keeps mom's blood away from baby's blood. But when are mom's blood and baby's blood going to meet? At birth. Mom's blood and baby's blood mix. Her immune system says, holy cow, did you know there was this foreign protein inside us the whole time? We didn't know it. Let's never let that happen again. Let's make a ton of memory cells towards that. If we ever see it again, we'll kill it. Mom gets pregnant a second time with an Rh positive baby. Her cells can't go over, but her antibody can. Antibody can pass through the placental barrier, goes after the baby's blood and destroys it. So that's what hemolytic disease of the newborn is. Not really a problem anymore because now we have RhoGAM shots. Okay, Rh gamma globulin, which is an anti-antibody. It goes into mom's blood. It ties up her anti-Rh antibodies. Now they're too big. Now they can't get across the placenta to get at the baby.
It's not called in the United States.
Exactly. That's exactly right. Yes. That's exactly right. It's not a problem in the United States. Type 3, still B-cell mediated. Type 3 is similar to type 2. You have a lot of IgG, a lot of IgM, some complement cascade going on. But what happens with type 3 is that you get huge antibody-antigen complexes. So whatever it's perceiving as foreign, it's sticking to, and it's creating these huge complexes. It's creating the complexes faster than your body can break them down. So these complexes begin to build up. They set themselves down, usually in epithelial basement membranes, and you have this constant inflammatory, kind of low-level inflammatory reaction because your immune system is inflaming in an effort to kind of break down these complexes, but you're building them as fast as you can tear them down. So you have this chronic low-level inflammation, and you get in trouble if that happens to be, let's say, your kidneys. That's a huge spot for epithelial basement membrane in your kidneys. If you inflame that, that's glomerular nephritis. Okay? Lupus is when it happens in the joints. So the joints become inflamed. So when you have these antibody-antigen complexes constantly building up, and then you have like neutrophils coming in, macrophages coming in an effort to clear those complexes, you'll inflame blood vessels, heart and lungs, joints, skin, kidneys, everywhere you have an epithelial basement membrane, If it's chronically inflamed, you run into a big problem. Type 4 is the only one that's T-cell. Type 1, 2, and 3 are B-cell mediated with antibodies. Type 4 is T-cell mediated. Type 4 is characterized by a latent period, usually a day or more between when you see the allergen and when you react to it. Infectious, it should say infectious, not infections. Infectious allergies fall under this category. Anybody know the infectious allergy without looking at your notes? Tuberculosis. That's the infectious allergy. Contact dermatitis, which is essentially poison ivy, is a good example of contact dermatitis, as well as graft rejection. Organ grafts, skin grafts fall under this category. Infectious allergies, tuberculosis. Okay? Tuberculosis test that a lot of people are given, they give you tuberculin, they inject it under your skin, and then you have to wait for your latent period. 1 or more days, and they'll ask you to come back in 24 to 48 hours, and they want to see if you have a reaction where they injected you. If you have a reaction where they injected you, that means that that particular bacteria has been in your body before, so you have antibodies towards it. If you have no reaction, it means you've never seen it before. It's infectious. If you have tuberculosis, that means everybody around you could possibly have tuberculosis, which is why you have to get it checked out. Now, can you have a false positive? Absolutely. If you have repeated tuberculin injections because they're constantly testing you, you could very well develop the antibodies just based on that. Anybody know what the second thing they would do to you to see if it was a false positive? Chest x-ray. They want to look in your lungs to see if you have the tubercles, okay, that are a symptom of having tuberculosis. Contact dermatitis is a delayed allergic reaction. The best example of this is poison ivy, but it can happen with many contactants, household chemicals, drugs. It usually also requires a sensitizing dose, just like a type 1 allergy, but again, this is T-cell mediated. Subsequent exposure to your allergen results in an inflammatory reaction that can severely damage the epidermis. So we're not talking about hives, we're talking about a humongous skin reaction, inflammatory reaction. From the initial contact until the time that you heal can take 10 days, which is significantly longer than a contactant allergy mediated by B cells. There's an example of contact dermatitis. Horrible damage to the epidermis. As people know that get poison ivy, if you scratch it, it gets worse and worse. You're actually doing the damage to your epidermis at that point, but you can scar yourself. Okay, graft rejection. Graft rejection is based on MHC differences. 2 types of graft rejection. Host versus graft, graft versus host. You kind of have to slow it down, and I'll show you a way to tell the difference. The graft is always whoever donated the tissue. The host is who's ever receiving the tissue. Host versus graft is an organ transplant, a heart transplant, a liver transplant, a kidney transplant. Those are all examples of host versus graft. The host is going to attack the graft. The first one in the verses is the attacker. In host versus graft, the host is attacking. In graft versus host, the graft is attacking. So host versus graft. Host T cells come out of the system. I just got a kidney from somebody else. They will eventually find the kidney. Macrophages will pick up dead kidney cells, sloughed off kidney cells. They will present it to my T cells. My T cells will immediately say, that is not from our body. That is foreign. We have to attack it. So they build up all their troops, all the T cells and B cells now go down and begin to attack my foreign kidney. The rejection process usually begins within 2 weeks of transplantation. They will eventually kill the graft without intervention. The quickest way to kill the graft is usually to shut down the vascular supply. The inflammatory response that comes with attacking a tissue usually inflames the vascular supply, therefore shutting off the blood to my new kidney. No more new kidney. So people that undergo transplants have to be on heavy immunosuppressive drugs slanted towards repressing the T cells, because the T cells are the ones most concerned with MHC. So here is host versus graft. Got a new heart. My cells come out. They begin to attack the heart. Graft versus host. There's only one way that you can get graft versus host. Bone marrow transplant. If the graft is going to attack you, that means you had to get somebody else's T cells and B cells. T cells and B cells come from the bone marrow. So if you get somebody else's bone marrow, you get their T cells and their B cells, which, since bone marrow generally only comes from adults, has already been trained and developed and differentiated in somebody else's body. So the new bone marrow, the graft, comes out of the bone marrow, and all of a sudden it says, I have no idea where I am. Everything is foreign around here, so I had just better go ahead and attack everything. So the effects of GVHD, graft versus host disease, are systemic. The entire body is attacked, and that's toxic. Usually, because your entire body is being attacked and there's only one organ that encompasses your entire body, your skin, the skin starts with a peeling rash, and that's the first sign of GVHD. 30% of people will reject about 100 to 300 days after the bone marrow transplantation, but that percent is declining because we're discovering more tissue markers. We're able to get a closer and closer match, but you'll never have a 100% match. I guess I shouldn't say never. Unless you have what? An identical twin. Types of grafts. Based on these words, I want you to figure out what the graft is. Autograft. What does auto mean? Self. This is like taking a piece of skin from the back of your leg and putting it on your arm. That's an autograft. It's coming from your body, just going to somewhere else on your body. Isograft. What does iso mean? It means same. So what's an isograft? Has to come from identical twin. The only way you could ever have an isograft is if you had an identical twin. Allograft. You want to take a guess here? Someone else of the same species. Allograft comes from another human. This is the most common type of transplant for organs. You take somebody else's kidney or somebody else's liver. So having put the of the same species differentiation, a xenograft is a different species. This would be taking a pig's valve out of its heart, or a cow's valve out of its heart, or something from another species and putting it into your body. Last thing, autoimmunity and underreactivity. So autoimmunity, again, is an overreaction. So it usually falls under a type 2 or type 3 hypersensitivity. Autoantibodies can be produced by B cells. T cells can sometimes cytotoxically recognize and try and kill self. It can be systemic, so you can have an overall autoimmunity, or it can be organ specific. Maybe it's just going after your joints, like rheumatoid arthritis. And here are some autoimmune diseases, where they fall in, what they're doing. Genetics and gender, autoimmunity clusters in families, which means that there's probably a genetic component, and there's a gender component, because females are more susceptible. We show autoimmune disorders more frequently than men, especially from puberty to menopause. If somebody has an autoimmune disorder, the symptoms are less before puberty and after menopause, but the age when they have high circulating hormones are usually the worst for their autoimmune disease, which means that there has to be some tie-in between the hormones and the autoimmune problems. Why do we have autoimmune diseases? Forbidden clones is one we already talked about. Clones got out and they shouldn't have. Sequestered antigen theory. When we talked about developing your B cells and T cells and showing them self, this theory says maybe there was some self we couldn't ever show them. The cornea of your eye is avascular. Blood doesn't get to it. Nutrients have to diffuse. Your central nervous system, blood-brain barrier, okay? These are tissues that your immune system can never see. So if something happens, if you have trauma to your eye or trauma to your central nervous system, and all of a sudden your immune system gets in there, whoa, what's this? It must be foreign because I've never seen it before. Let's attack it. Okay, so that's sequestered antigen theory, or not your cornea, your lens. My fault. Immune deficiency says that the receptor's mutated. What was a specific receptor mutated, and all of a sudden its mutation is for self. Inappropriate expression of MHC2. Things that overexpress MHC2 or underexpress MHC2 trigger your immune system. Mostly because some cancers overexpress MHC2 and some viruses think that they're so smart when they're hiding in cells, they pull down that cell's MHC2 because they think they're hiding. Your immune system's on to that. So if it sees inappropriate expression of MHC2, it automatically kills whatever cell is expressing it. And last one, molecular mimicry. This supposes that there are substances that exist that are so close to being you that if they enter into your body, they look enough like you that when they're taken care of by the immune system, they're perceived as foreign, they're eliminated, and then your body turns on itself and says, that looks a lot like what we just got rid of. Let's just be safe and attack that too. So that's molecular mimicry. Last thing, immunodeficiency, hyposensitivity, and underreaction. Two categories, primary and secondary. Primary means it was present at birth. Secondary means it was an acquired immunodeficiency. I do want you to know this chart for your final exam, and here's how to read it. Over here on the left, you have the bone marrow. Over here on the right, you have what would be taken care of by a fully functioning T cell on the top and B cell on the bottom. So, we have our bone marrow. It makes a lymphoid stem cell. X, if it cannot make a lymphoid stem cell, if your bone marrow is unable to go from the pluripotent stem cell to a lymphoid stem cell, that's called SCID, S-C-I-D, severe combined immunodeficiency. Means you have no T cells, no B cells. Okay, we'll do T cells first. We have our bone marrow. We have our lymphoid stem cell. It made a pre-T cell, but the pre-T cells would not migrate to the thymus. We have an X going to the thymus. That's called DeGeorge syndrome. So DeGeorge syndrome is the failure of T cells to come out of the bone marrow and go to the thymus. Okay, so start over again. Bone marrow, lymphoid stem cell, we have pre-T cells. They went to the thymus. They developed and differentiated, but they will not activate. We show them their specific antigen constantly. They won't activate. This is called ADA, adenosine deaminase deficiency. Adenosine deaminase is an enzyme that T cells have to have in order to activate. Okay, so if they are mature and they are specific, and we show them their specific antigen and they won't react, that's ADA, adenosine deaminase deficiency. If any of these occurs, you will have recurrent fungal, protozoan, and viral infections because those are the foreign invaders that T cells mostly attack. B cells. We have our lymphoid stem cell. We get a pre-B cell, but it will not mature inside the bone marrow. Okay, that X, if we have a pre-B cell but it won't mature, that's called congenital A-gamma globulinemia. Okay? A gamma, that means the opposite. That means it will not make its immunoglobulin, which is its B cell receptor. It won't mature and take on specificity. Bone marrow, lymphoid stem cell, pre-B cell, it matures. It comes out into the circulation. It will not produce antibody, even when shown its specific antigen. This could also be ADA deficiency in the B cell, or it could be called hypogammaglobulinemia, which means it might be producing antibody, but at a very low number, at a not an effective number. If your B cells are not functioning, you're going to get recurrent bacterial infections, because that's mostly what the B cells battle are, the bacteria. In some cases, okay, if you have pre-T cells and pre-B cells, but they won't mature, that's called X-linked SCID, severe combined immunodeficiency. It simply means it's another type of immunodeficiency carried on the X chromosome, passed from mother to son. There's more immunodeficiencies. Last slide. Taken way more time than I wanted to. I apologize. I can stay longer for anybody that wants additional time. You don't have to exit at 11. Secondary deficiencies caused by one of 4 general agents: infection, organic disease, chemotherapy, and radiation. The AIDS virus. If there was one cell type that we've talked about in innate and adaptive, if there's one cell type that you could target to take out your entire immune system, which one would it be? T cells. T cells are the cornerstone of your immune system, and that's what the AIDS virus goes after. Once it successfully knocks out your T cells, that's when HIV turns into AIDS. When your immune system has been compromised, the human immunodeficiency virus has caused you to develop acquired immunodeficiency syndrome. That's when HIV changes to AIDS. Organic disease, cancers, especially leukemias, okay, leuko going after the white blood cells, creating a problem in the white blood cells. Chemotherapy and radiation, what do they target? Cancers, but there's a more generic way to say what they target. A rapidly dividing cell. That's all you can say about chemotherapy and radiation. They're really good at killing rapidly dividing cells. Cancer, yes, is a rapidly dividing cell, so they are very good at killing cancer. But you know what else rapidly divides? Your bone marrow. So you cannot receive chemotherapy and radiation, systemically anyways, even locally sometimes, without also putting a dent in your bone marrow. So if you receive systemic chemotherapy or radiation, you put a dent in your bone marrow, which knocks down your blood cells and your immune cells, and that makes you immunocompromised. Questions? Okay. Hold on.